game, that Clemson-Carolina game? Who's going to win that game, huh? How many Clemsonites do we have in here? Oh, isn't that great? Isn't that great? That's wonderful. That's wonderful. Truly is God's country, isn't it, huh? Beautiful, beautiful part of South Carolina. But, you know, this is a beautiful part, too, and I really miss it here. Many, many good years, good friends, good associations. So, today, we're going to talk about a group of viruses that aren't as interesting as the group of viruses we're going to talk about tomorrow, the herpes viruses. We're going to get into sex tomorrow, talk about sexually transmitted diseases and all that good stuff. And I know you're going to be interested in tomorrow's lecture, probably a little bit more than today's lecture. But let me try to make today's lecture a little bit interesting, because this is probably ranks at as one of the top group of viruses that you're going to see out in practice. And for the most part, they're going to be undiagnosed. I mean, people are going to have febrile illness, rash. The kids are going to be infected with Coxsackie viruses. There's going to be respiratory infections with the echoviruses. But you'll never know it, because you'll never go to the trouble of trying to identify what those viruses are. But they are a leading cause of human illness, and they are fecal-oral transmission. And those are things that you need to know about this group of viruses. So let's get into them. I just wanted to put this. I love this group slide here. You know, here I am here. I had a lot of hair back then. And here's Dr. Mayer, and you can see how much he's aged over the years. I always like to show this, just to keep things in perspective. You know, what was so interesting? When I was here, they used to get me mixed up with this guy here. Gafar, Gangemi, you know, they just couldn't get that one straight. I don't know what it was, but yes, that's Dr. Gafar right there, huh? Who's the best looking one out of that? Oh, boy. Let me tell you, this group is ready. This group is ready. Okay, so here's how I like to break down the lectures, and I'll do the same thing with tomorrow's lectures. We've got 2 hours tomorrow to talk about the herpes viruses. But with the enteroviruses, go through a little bit of the background on the viruses, how they're classified, basic biological properties of the virus, talk about pathogenesis, immunity, and then disease. All of this in 50 minutes. And Dr. Mayer said, I have exactly 50 minutes, and I got to get off the stage. So let's see if we can do it. Okay. This group of viruses is part of the large virus family called Picornaviridae. Pico, small, RNA, family of viruses, Viridae, right? I know Dr. Margaret Hunt has told you about some of the other virus families, but this Viridae, family of small RNA viruses. Okay, so they're all RNA viruses. Now, there are 5 genera in this group, and we've got the rhinoviruses, which cause upper respiratory tract infections. There's over 100 serotypes. We really don't have time to get into it. So rather unfortunate, but many of the upper respiratory tract infections that you're seeing this time of year are due to this group of viruses. Another group that we don't talk too much about, although one virus in the genus Heparinavirus is the virus which causes infectious hepatitis, the hepatitis A virus, and that is included on your lectures in hepatitis. And so it's part of this small RNA family of viruses, but we're not going to talk about them at this lecture. 2 other groups, the cardiovirus and the aptovirus. Again, these are small RNA viruses associated with some human disease, but very limited. And so we don't go into them in much detail at all. So let's take the one genus that we do spend, and this lecture is going to focus on. It's the enteroviruses. And there are really 3 groups in the enteroviruses: the echoviruses, the Coxsackie viruses, and the polioviruses. Coxsackie virus, do we have any Yankees in here? We got some Yankees? Where's my other Yankee in here? Where are you from? New Hampshire. Okay. New Hampshire. Do you know where Coxsackie, New York is? Okay, Coxsackie, New York. That's where this virus group was actually, this virus was isolated officially from Coxsackie, New York. The echoviruses, the enteric orphan, cytopathic orphan viruses, rather unusual name, but it was given because they didn't know what else to call them. And so that's a third group of viruses. But the hallmark, or the virus that is really the prototypical virus in the enterovirus group, is poliovirus. And we're going to talk a little bit about poliovirus and the disease polio. Okay. Dr. Hunt has told you about these viruses, but I just want to give you a quick reminder. These are single-stranded, positive strand RNA. The RNA, the genome, is infectious. On the 3' end, it's got a poly-A tail, and on the 5' end, it's something called VPG. Now, VPG is a small protein that is required for the initiation of infection. The capsid has 4 polypeptides associated with it. This is an icosahedron virus. And remember, there are 12 vertices with an icosahedron, VP1 through VP4, 4 distinct proteins that are associated with the capsomeres of the virus to form the capsid. The size, again, it's a very small virus. It's only about 20 to 30 nanometers in diameter. Now, tomorrow when we talk about the herpes, to put things in perspective, we're going to talk about viruses that are at least 3 times the size, 4 times the size of this virus. This is a very small virus. It has a naked capsid. There's no envelope associated with it. And that, as we're going to see, has a lot to do with its ability to cause disease and the type of disease that it'll cause. Now, here's just an electron micrograph. This is a negative stain of purified viruses that were grown in cell culture systems. And you can see that they line up very nicely and can form a crystalline array. And actually, when you look at infected cells, sometimes you'll see in the cytoplasm this crystalline array of virions that can be produced. If you look at fecal samples, this virus and all the viruses in the enteroviruses grow to very large titers, high titers, and are excreted in the stool in very large quantities. And again, that is one of the reasons, together with its ability to not break down following high pH or high temperature environments in the intestinal tract, but the large amount of virus that's there gives it the ability to be transmitted by the fecal-oral route. Again, that's one of the key points with this group of viruses. And so if you take apart the virus and look at it in just a little bit of detail, again, here's the negative stain of the virus, the icosahedron. And again, remember, you've got the 12 vertices, the pentons, and you've got these 4 polypeptides, VP1, 2, 3, and 4. And I'd like you to remember VP1 as being a very critical component of the virus, because there is an area within VP1 that has a so-called canyon area associated with it. And that's a hidden area on the capsid shell, which is required now for uncoating of the virus. We'll come back to that in just a minute. But VP1 is actually the driver of this virus. These viruses cause a variety of different infections in different tissues in the body. And it's VP1 that really drives the virus to specific target sites throughout the body. Okay? Now here's a blow-up of a diagram of what we're talking about here. This is a freeze fracture of the virus. This is a computer-generated reconstruction of the virus. But what I'd like you to focus on is this VP1 and VP4 region, okay? Remember that you've got this component, the four polypeptides coming together to form the capsomere. Here is the canyon of the intersection between the VP1 and the VP4 region. This is a very important region because this is the region that recognizes intracellular adhesion molecule 1, ICAM1. The recognition of ICAM1 from a cell into that canyon region is required now for this initial attachment and the uncoding of the virus. Okay? It's a very important concept. Most enteroviruses have this relationship with ICAM1. The poliovirus is a little bit different. It doesn't require the ICAM1 relationship to get into a cell. And instead, it requires other cellular adhesion molecules on neurons, okay, to get in, and that's why we get paralytic polio disease occurring. Just to summarize, VP1 is the key region on the vertices and on the capsid itself, allowing for the virus to attach to a susceptible cell and initiate infection. ICAM1 is the cellular component, the cellular receptor, that's responsible for most of the enteroviruses getting in. When we come to polio, we'll talk about some other molecules that are also involved. I don't think I have to tell you anything about this because Dr. Hunt has told you basically about the replication of the RNA viruses. But of course, the RNA viruses replicate in the cytoplasm. And with polio, you get these large crystalline arrays of virions. Now, poliovirus is rather unique in that it actually blows away the cell. In other words, when it infects, it very quickly is able to overtake the cell machinery. And some of the proteins in the capsid shell will actually turn off the ability of the cell to go through its normal replication process. And so the RNA of the virus and these proteins take over the cell, and the cell is not able to make its proteins. Translation doesn't occur at the normal rate. And within a very few hours, the cell completely is taken over by the poliovirus and by some of the other enteroviruses, and it'll die. But before it dies, you have this tremendous bursting effect. And so this cytopathic effect, this lytic effect, occurs. In the enteroviruses, it is the lytic disease that's most important. Tomorrow we're going to talk about immunopathogenesis and the immune response to the herpes viruses, which are largely responsible for the disease characteristics. This virus, this group of viruses, these are lytic viruses, and it's the breakdown, the complete destruction of the cell that's responsible for the diseases that we're going to be seeing. Okay, so what are these diseases now? What do we have in this group of viruses? There's a number of them. And the disease is in part due to VP1. Okay? Because VP1 on the echoviruses, the Coxsackie virus, and the poliovirus are slightly different, driving it to different tissues, causing different diseases. Okay? And we're going to talk about how the virus gets in and how it's disseminated. But here's basically 8 different diseases that can be caused by the enteroviruses. Just a non-specific febrile illness this time of year. You'll see kids who are infected with the Coxsackie viruses, and they have fever and they're lethargic. You can't diagnose it. It's not worth diagnosing because they recover in a very short period of time. Perinatal infection with Coxsackie viruses can be very severe, but you don't see that very often. But Coxsackie B can cause an effect in young infants. Febrile disease with rash, okay? And you see that, again, a lot this time of year. It's like nonspecific febrile illness, but now there's a rash associated with it. And both Coxsackie A and Coxsackie B viruses will cause that. Meningitis, the ability to infect the meningeal cells, the ability to infect heart cells. All of the enteroviruses basically have this capability, But we usually see it with the Coxsackie viruses, and usually it's Coxsackie B viruses that cause that. Pleurodynia is a disease that we'll talk about in just a little bit of detail. It's the grip, the devil's grip. Anybody have an aunt or uncle that have ever talked about the devil's grip? Never heard about that one. The devil's grip. Well, you can imagine what it is. It's like somebody having a large hand and just squeezing your chest. Very much like a coronary, so people say, and many people go to an emergency room complaining of this sharp pain and this gripping across their chest. The intercostal muscles are affected. Is the devil's grip, that's the name given by the old-timers, but what it actually is Coxsackie B virus infecting those cells, okay, the intercostal cells, causing this destruction of tissue there, and causing a syndrome that appears to be like a coronary attack, but it's not. The last one, poliomyelitis. The polioviruses, and they're serotypes 1, 2, and 3. Most poliovirus is caused by serotype 1. Most people who are infected with the polioviruses, if you look back at the literature in the 1950s and in the 60s, most people who were infected had so-called silent disease. What's a silent disease? Silent disease? What is it? No symptoms, right, no symptoms. And really this is, again, another hallmark for most of these viruses. They infect a lot of us, but they don't cause any disease, no symptomatology. Things have to be just right. You've got to have the right amount of virus coming into your body through the right portal of entry. Got to be the right age, seronegative and whatnot, and then you can be infected and have clinical disease. But most of us here have antibodies to echoviruses, Coxsackie viruses, and even the polioviruses, those of us who weren't vaccinated with the poliovirus vaccine, that is. And so silent infections, the ability to be infected but not have clinical disease, is really a hallmark with this group of viruses. Okay, so those are the 8 general characteristics and the 8 general diseases that we're going to quickly talk about. All right, a little bit of the background about these viruses. It's as Dr. Mayer will tell you, I like to talk about the history because I like the old-timey stuff, and I like to look back at what really happened with this group of viruses. But really, this group of viruses gave rise to modern-day virology for a number of reasons. One is that Salk and Sabin back in the 50s were the first to actually isolate wild-type virus and pass it through cell culture systems and attenuate the virus. What's attenuation mean? Attenuate the virus. Less virulent, right? They were the first ones to be able, with the polio virus, to do this. Now, before them, of course, you had other people looked at this, Pasteur and Jenner, and they did things. But that was many years prior to Salk and Sabin, who in the 50s were able to attenuate the poliovirus and come up with a poliovirus vaccine. And of course, we've got the killed vaccine, the Salk vaccine, and the Sabin, the live virus vaccine that's out there. And I think Dr. Richard Hunt's going to talk with you about the vaccines, the Salk and Sabin. But here's a couple of guys that don't get much credit. You don't hear much about Enders and Weller. They were the guys who were really able to perfect cell culture. Now you might say, well, what's so neat about that, or what's so interesting about that? Well, if you can't grow these viruses in cell culture, if you've got to use animals all the time, you can't do the sophisticated types of things that you'd like to be able to do with the viruses. And so Enders and Weller were the two guys that really perfected the cell culture techniques, working with both Salk and Sabin to grow the virus in cell culture. Both of them ultimately won the Nobel Prize. All of these were Nobel Prize winners. So this virus was really the beginning of modern-day virology. Okay, some newer people that have come into the picture. Dr. Sharp, who did some very elegant work on the replication of RNA viruses, and again was able to add to the literature on mechanisms of RNA and how RNA is replicated and the translation of RNA. He actually used the virus simply as a tool to study modern-day cell biological effects. X-ray crystallographic characterization, fine structure mapping, all of this was done on this group of viruses, the enteroviruses. Okay, we've already talked about this, about their size, about the capsid and the role of ICAM1. But let's talk about why these viruses are so infectious now. These are naked viruses. They have no envelope. If a virus has an envelope, physically, what can it do, what can it do? The virus has an envelope that's got a lipid that surrounds it. Fuse the cell membrane? Yeah, good one, good one, good one. How about its ability? How about its hardiness? Get back to its hardiness. There, which one? Which one is more hardy? Is more hardy? You think so? Good. Great, great, great. Okay, so naked viruses are the hardiest, usually, in the virus world, because they don't have this envelope which can be deteriorated by UV light, by drying, by pH conditions sometimes can break down the envelope. And so naked viruses don't need an envelope, and so therefore the integrity of the virus is contained in its capsid, in this protein shell. And the enteroviruses are very hardy viruses. They withstand low pH, pH 3, okay? And they are, God bless you, they are able to, there's some virus in that sneeze, you know. So they are able to withstand the pH of the stomach, the enzymes in the stomach, in the gut. They are able to survive the harsh body environment as well as the external environment of sunlight. That they don't have to worry about being dried out. They're very hardy viruses. That, coupled with the fact that they grow to very large concentrations and are excreted in very high concentrations in stool, makes them highly transmissible viruses. And so that's really key to this group of viruses. And by the way, I might say that some of the other viruses in the picornaviride family are distinguished on their pH sensitivity. For instance, the rhinoviruses, they don't have quite as good a pH ability to withstand low pHs, okay? It's not quite as good. And the rhinoviruses need to replicate at temperatures less than the enteroviruses. And so when you think of the rhinoviruses, they're replicating in the nasal passages and the upper respiratory tract, the temperature is less, it's not as high as the temperature in the gut. And so that is a distinguishing feature with the rhinoviruses and the enteroviruses. But these viruses are able to stay around in feces, stay around on tabletops for long periods of time and retain their infectivity. And that's what's important with them. And one of your handouts simply has, just shows what we've said about their ability to remain stable at acid pH. And if we look at poliovirus, there's 3 types of poliovirus. There's a number of types of Coxsackie A and B. I don't expect you to know those exact numbers, so don't worry about that when you're studying for my test questions. That's not important. I do want you to know some of the diseases that are caused by Coxsackie A and B and the echoviruses, but the actual identifications number-wise are not important. Okay. So, we've covered a lot of this, but fecal-oral routes is important. They grow in tissue culture very well, many times without CPE. They cause silent infections. okay, as well as a number of important illnesses. But this part right here, silent infections with this group of viruses. All of us have been exposed to a variety of them. All of us have antibodies to them now without ever having any knowledge that we were infected with the viruses. Okay. Let's get into pathogenesis now. Let's talk about pathogenesis and what happens with this group of viruses. For the most part, with these viruses, they enter via the mouth. And they can either be in a respiratory droplet, or they can be in contaminated water, or in contaminated food. Okay? Now, the key thing to remember, and you're going to see a diagram on the next page, which really shows this in detail, is that the viruses come into the body, and they need to go through an initial round of replication, an amplification stage. And they actually replicate in the oropharynx and in cells, the mucosal that surrounds the oropharynx. They replicate to rather high concentrations in those cells, and initially there's a slight fever that's associated with that high replication and perhaps a sore throat in some individuals. But the virus then is swallowed. Okay? The virus is swallowed. And the real important stage then is the swallowing of the virus, the ability of the virus to survive the high pH, the high temperature, and the enzymes of the gut. Because of that capsid structure, it survives, can infect mucosal cell surfaces in the gut. It gets over into Peyer's patches, the draining lymph nodes in Peyer's patches, replicates very high to high quantities in there, and then gets into the blood. And this is really the key part of infection with these viruses. Because once it causes a viremia, you have a seeding of the virus to the target organs, depending on the capsid, depending on VP1 and that interaction, and the disease that's then going to ensue. And let's look at that. Let's look at that in some detail here. The virus gets in. Okay, rhinoviruses need to stay up here because pH is too harsh on it down here. Temperature is too high. The rhinoviruses will stay up here, causing the common cold. The enteroviruses, however, they withstand the high pH, high temperature, and enzymatic conditions in the stomach. Okay, first round of replication, oropharynx. Okay, and then they replicate within the gut, causing this primary viremia that I've just talked about. It's VP1 now that then drives the virus to different target tissues. And so if we're looking at different organs, skin, muscle, brain, meninges, liver, we can see that what happens is that, for instance, in the skin, the Coxsackie A virus has a predilection, because of VP1, to get to the skin cells and to cause rash and also these papules that I'm going to show you in just a minute. In the case of pleurodenia, it's Coxsackie B virus predominantly that gets to the intercostal muscles and those cells, infects those cells because of that interaction with the cell surface in VP1, and causes pleurodenia, devil's grip. In the case of poliovirus now, here's the exception. Poliovirus recognizes a slightly different cellular receptor. It recognizes the cellular receptor not only on muscle cells, but receptors that are present as well on neurons. As we're going to see, the initiation of poliomyelitis is an infection of muscle cells followed by an infection of the innervating nerves that are associated with those muscle cells, and the virus then killing those neurons, causing poliomyelitis. In the case of infections with the meninges, there are other viruses that can cause a paralytic-like disease or a meningitis. As a matter of fact, each of the three groups, Echo, Coxsackie, and polio, can infect and cause a meningitis. In the case of the liver, we've already talked about the parnavirus, the infectious hepatitis A virus, which is now classified in a separate group, but it has the ability to recognize those receptors on liver cells and cause hepatitis. But again, to put this all in perspective, the virus comes in the mouth, the ability to survive in the intestinal tract, cause of viremia, and it's that viremia then that pushes the virus out throughout different organ systems, depending on VP1. That's the disease that you get. Okay? Now, any questions? That was a lot of information. Most of that's all in your notes. I think everything is in your notes. But anybody have any questions on what we've covered so far? Okay, let's just talk then about what's really happening. Let's take a deep breath here and slow down a little bit. Most of these viruses are, again, fecal-oral transmission, okay? So most of us have good hygiene, I hope, right? Got a good hygiene, and so we're not really spreading the virus around too much. But where is the virus being spread around in great quantities? Oh, boy. Who's worked in a daycare center? Who's worked in a daycare center, huh? What do you see in a daycare center? I got 3 grandkids, and I've been to the daycare center. And, you go into that one little area in the daycare center where there's a couple girls there, and there's an assembly line, they got the diapers and, quick wipe, and then throw that diaper into that pail. You can see the virus just coming out of that. You can just see it coming out of that. That's where that virus is. That virus is in that fecal material. And kids do get infected, particularly with the Coxsackie viruses. And they get this febrile illness, sometimes with rash. But their feces is loaded with this virus. It's easy to go in. It's easy to find this virus. But how about us? I mean, what's in areas of better hygiene? How many oyster eaters do we have here? What? Huh? We got some oyster eaters. When is that next oyster roast? The oyster feast in Charleston at the Sullivan's Island is one coming up, I think, isn't it? You love oysters? Everybody love raw oysters? I can see some of these others saying no. Raw oysters, okay? All right, so I'm a big oyster eater, and I love oysters. Now, if you have the oyster roast, you can pretty much inactivate any virus that's there. But I like raw oysters. Well, I did a little survey a few years back and looked at my sera as well as a few others who were oyster eaters. What do you think I found? Man, scary. Scary. I mean, antibodies to just about everything that you can imagine. But I never got sick. I never got sick from eating raw oysters. Now, probably because a silent infection. There wasn't enough virus in that oyster to actually make me sick, thank goodness. And now I've probably got good immunity, so I could eat contaminated oysters and, be fine. How many golfers do we have? Got a couple golfers? All right, guys, I used to play golf at one time, but, in the summertime particularly, you want to get an early start. You want that 7 o'clock start out on the tee, and you call and you say, give me a tee time, 7 o'clock. And you get out there, and on the coast, play along the coast. Boy, you get out there, and man, it's the most wonderful thing. The sun's just coming up, and they've just irrigated the tees and the fairways. Take that deep breath. Boy, isn't this wonderful to be out here? What do you think you're inhaling out there on that golf course? Yeah. Who said that? Yeah. Well, it's not actually sewage, but along the coast, what do you think that the owners of these golf links have a special deal with for irrigation? Yeah, they do. And there is a water that you can get that is not potable water, okay, not fit for drinking, but is fit for irrigation, okay? Well, it hasn't gone through the tertiary step that's required to be potable, okay, to be drinkable, all right? And so what do you think's in that water? All those viruses that we've talked about. All of these enteroviruses can withstand sewage treatment, again, because of that novel structure of that capsid shell, the virus. It can withstand sewage treatment, and hey, it gets through, and so, yeah, you're out there and playing golf, and you take that nice deep breath, and that nice mist that's out there. That's what's happening. Many of you haven't gotten sick, but maybe some of you have if you're in the wrong place at the wrong time. Okay. By the way, also worked with a guy that did actually, he looked at people that worked in sewage treatment plants and did the same analysis of that. This was down in Texas, in Galveston, Texas, that they did this. They have a big water virology group in Galveston, Texas. What do you think they found with the sewage treatment plant workers? And they've got antibodies to everything. Everything. Surprisingly enough, though, not a lot of disease that they could really track down, unusual disease, but they've got antibodies to all these agents. So, ubiquitous viruses, they're all over, they're everywhere. Silent infections for the most part. Okay, immunity. Dr. Mayer has told you about immunity to viruses and to bacteria, and I'm sure Dr. Mayer used the military analogy with the bombers. And has he used that? Did he go through that? He didn't. He doesn't. His lectures are so dry. I know. I know. What can I say? So in immunity, for this group of viruses, remember, disease is primarily cytopathic effects. The virus blows the cells away, okay? Whereas other viruses that hangs on, and it's the immune response to the virus that's the primary cause of disease. In this case, it's humoral antibody that's most important in resistance to disease. And in the Salk and Sabin vaccines, it's the generation of a neutralizing antibody, which can prevent poliovirus from infecting. Okay. Again, we've talked about these characteristics in your notes. Let's just talk a little bit about the diseases. Aseptic meningitis, and you'll see that quite often. And the cause of that can be several different viruses, as I've told you. But it can also be bacteria can cause aseptic meningitis. Chlamydia, leptospira can cause it. Perhaps the best way to differentiate is to look at CSF. And of course, if you find polys in there, if you find bacteria in there, you know it's not a virus. For most aseptic meningitis cases, it is usually a self-limiting disease and will go away. A number of viruses in this group will cause aseptic meningitis. You won't go through the identification of it because it's too difficult to do, and hopefully your patients will recover within a few days after the meningitis starts. But let's look at poliovirus, and let me just tell you a little bit about poliovirus. In the 50s, when poliovirus was really being evaluated, studied in great detail, what was known was that usually the polio caused poliomyelitis, not in very young kids, but in kids that were 8 to 10, and in some cases in adolescents. Young kids were pretty much resistant to being infected with the virus. And as it turns out, probably less than 0.1% of the people who were infected with the poliovirus actually came down with poliomyelitis. And to get into some of the details, you had to have the right serotype. Serotype 1 was the one that usually caused poliomyelitis, although 2 and 3 could also cause it. But you had to have a big bolus. I'm talking about the oysters. You had to have a lot of virus come into your body. It was thought to actually cause an infection which would result in the severe poliomyelitis syndrome. Okay? So only a few, though, that were infected actually came down with polio. But if you looked at sera in kids from the 1950s, almost 90% of the kids in many areas had antibodies to either type 1, type 2, or type 3 poliovirus, but didn't have the poliomyelitis. Again, the virus entered through the oropharynx, causes a viremia, and about 7 to 8 days later, it's seeded to the CNS, and you could get a major illness, which could be a meningitis. It could progress, and you could develop a paralysis. Now, let me tell you about the different cases with poliovirus. So, some of the kids that were infected developed meningitis, and then they recovered. Some of the kids that were infected developed meningitis as well as a paralysis. And it could have been a flaccid paralysis in which some of the motor neurons that innervated various muscles of the legs or the arms were also infected, okay? Depending on the number of neurons that were involved, paralysis was either permanent or there was a recovery. If you only had a few of the neurons infected, you could recover from flaccid poliomyelitis, okay? From flaccid poliomyelitis. However, an even smaller number of kids developed something called bulbar polio, in which a large number of neurons in these motor regions were infected and were killed. They were lysed. The neurons were just destroyed. And if the virus then went to even further into the CNS and was able to infect respiratory centers in the CNS, you got something called bulbar polio, in which they couldn't contract their muscles for their lungs to breathe, and they needed to be put into an iron lung. Thank goodness none of you probably have seen an iron lung, and hopefully no one will see an iron lung. But an iron lung was used to compress the chest so that you could breathe and life could continue. But also the saliva that would form because you couldn't swallow. And you can imagine these young kids who couldn't breathe, they couldn't swallow. It was a very terrible disease, and that was bulbar polio. Okay? Only a small number of kids actually came down with bulbar, although when you see these iron lungs and see these wards full, it's not all that insignificant. Now, today, we do see in some people something called post-paralytic syndrome. Post-paralytic syndrome. I'll get to you. Let me just get through this one point. Post-paralytic syndrome are those people who came down with polio and recovered. Okay? They recovered, all right? Because the neurons, all the neurons in their motor functions were not destroyed. And so the neurons actually recovered, and so they recovered use of their arms and legs. But as they aged, they lost, naturally, neuron function, okay? And so we talk about senile dementia. And, there's this aging, associated with aging is this gradual loss of neurons, theoretically, that occurs in all of us, okay? And so if you're at a deficit when you get polio, then as you age, some of those motor neurons, okay, can't compensate. And now you get post-paralytic polio syndrome. And a few years ago, this was a hot topic because a number of people who had polio recovered from it now were showing similar signs of the original poliovirus disease. Okay? But post-paralytic polio, those neurons that were knocked out early on and the aging process results in this syndrome that looks very much like the original polio. Okay, that summarizes everything that I've said there. Again, the spinal or flaccid form with the extremities. But here's bulbar, and again, we've talked about bulbar, and it involves the cranial nerves, respiratory and circulatory centers. We've talked about the post-polio syndrome right there. Question?
I just wanted to ask
if the kids in the iron lungs recovered, usually not. Usually not, because there were so many neurons knocked out, okay, that unfortunately it was a terrible thing to be in an iron lung, of course, as you can imagine, for the rest of your life. And many of them were. Many of them were. Those of you going to Washington, if you go to the Museum of Natural History in Washington, there's a wonderful expose on poliovirus there. Go take a look at that. They even show some of the iron lungs, and they talk a little bit about the history of poliovirus. Okay, now I'm not going to get into Salk and Sabin because Dr. Richard Hunt gives a great lecture, and he tells you about the differences in the two vaccines. I would like you to remember, though, that the Sabin is the attenuated vaccine, and the Salk is the killed vaccine. And one of the reasons that we've gotten away from the live attenuated vaccine is this reversion. That is, some of the kids who were vaccinated with this live virus vaccine, which is a great vaccine, actually would excrete virus that was the wild-type virus. And some of the kids that received the vaccine actually came down with poliovirus. A very small number, but still it was enough for FDA to say, hey, let's get out of this. Let's get back to the less effective killed vaccine. That is the Salk vaccine, which you don't have these problems, and let's avoid any complications. And Dr. Richard Hunt's going to tell you about the complications and the differences between those two, so I'm not going to spend a lot of time with that. Okay, now here we're going to close out, and we're going to talk about diseases really with the Coxsackie viruses, and I'm just going to pick out a couple of them that I'd like you to focus on. One is herpangina, a very unfortunate name. Because when you hear herpangina, what do you think about? Herpes viruses. You think about herpes. Herpangina causes a mouth ulceration that is very similar to gingiva stomatitis, which is, in fact, caused by herpes viruses. But herpangina occurs very frequently in kids, and it is caused by Coxsackie, mainly by Coxsackie A viruses. And there's a characteristic lesion that's formed in the back of the mouth, and I'll show you some of those lesions in just a minute. I've told you about pleurodinea, and not as uncommon as you might think. Pleurodinea, devil's grip, occurs quite often, and those of you who go into emergency medicine are going to see this quite often. And again, it's not a coronary occlusion, but it is this intercostal muscles that are infected by this group of viruses. God bless you. Aseptic meningitis, we've talked about that. We've talked a little bit about neonatal disease that can be caused by mostly the group B viruses. Respiratory infections, I've told you that there are a number of respiratory infections caused by the Coxsackie viruses. No need to try to identify them. Very mild respiratory disease. It comes and it goes. Now, just a little bit about hand, foot, and mouth, like herpangina. Again, kids, it's scary because Johnny will come home after school and he'll say, Mama, I don't feel well. My throat's sore. I've got a rash and I've got a fever. And Mama looks at Johnny and looks at his hands, and all of a sudden there are all these bumps, all these papules on his hands. It looks like smallpox, actually, the beginning of smallpox, these papules that form. And on the soles of the feet, again, these papules that'll form. And Mama gets very upset, of course, and goes to the family physician. And, of course, you'll be there and you'll say, nothing to worry about. That's hand, foot, and mouth disease. That's Coxsackie virus. Within a few days, that'll go away. And it does. It resolves very, very quickly. But in this case, it's a Coxsackie A virus causing these vesicular lesions. Now, a disease that's much more significant is myocardiopathy caused by the Coxsackie B viruses together with sudden onset diabetes. A lot of research going into these two syndromes. The Coxsackie viruses are believed to be the principal cause of this cardiopathy. And the Coxsackie B viruses are believed to play a very important role in sudden onset diabetes as well. And there's nothing really you can do about it. Again, these viruses are ubiquitous. Some people have genetic predisposition to come down with myocardiopathy or the sudden onset diabetes after infection with the Coxsackie viruses. Here's the picture that I wanted to show you. The uvea, you can see at the back, the tonsillar crypts back here. You can see these lesions that form. This is herpangina. Here's the lesions that occur on the fingers, and they also occur on the palms of the feet. This is hand, foot, and mouth disease. This will give you a good, Dr. Mayer, would you believe I am going to end this on time? This is, your clock is 3 minutes fast up there. 3 minutes fast. Okay, this really brings it all together for you. All right. So here are the diseases that we've talked about, and you know I want you to know, what is this virus? Where is it? Why does it cause the diseases that it causes? Okay, and what are the diseases? And here they are. And you can see this list of different diseases. We've covered most of them. You know, I'd like you to look at the common ones and just know the common ones, okay? Know the common diseases and know the virus that's associated with them, whether it's a Coxsackie-A virus. Coxsackie-A virus, visceral type diseases in the mouth. Coxsackie-B, internal body. Remember, body is the B virus infections. We didn't talk about echoviruses because they're just not that significant. They do cause disease, but you don't need to burden yourself that much with them. I do want you to know the diseases associated with Coxsackie, the A and the B, as well as the polio virus and how the polio virus gets in and the enteroviruses in general get in, how they're shed and how they spread from person to person. But more importantly, why do they cause the disease that they are associated with? And I think, yeah, there's one more table in the back that, again, is from your book. By the way, I think this is a great book. How many of you bought this book? Good for you all. Good for you all. Our Clemson grads bought the book. You know, Dr. Margaret Hunt, Dr. Mayer, do such a wonderful job for you guys. I mean, you know, who else would put these on the web and have these notes, these wonderful notes handed out to you? They do a wonderful job. But every once in a while, go back and just take a look at this book, because there are some hidden things that we can't say, we don't have time to say, that are in the book. Use your notes as a guide, but go back and use this as a reference to really become experts in the area.